The bulk of today's lesson is actually going to go over the design of experiments. If you've taken any science classes, I'm assuming a lot of this will sound familiar to you, but it is the same idea in data collection and in statistics, since this is usually the conclusion piece to any scientific study anyways. Definition-wise, an experiment is a controlled study conducted to determine the effect that varying one or more explanatory variables or factors has on a response variable. Remember, response variable is what's changing based on our treatment. It's what we're measuring, what we're studying. Any combination of the values of the factors is called a treatment. Even if it's not like a medical treatment, we're still going to call them treatments. In an experiment, an experimental unit is a person, object, or some other well-defined item on which a treatment is applied. We often refer to the experimental unit as a subject when it's an actual person. When it's like a plant, you would call it the experimental unit. The goal in an experiment is to determine the various effect treatments have on the response variable. So for example, if we want to determine whether a new treatment of some sort is superior to an existing treatment or no treatment at all, to make this determination, experiments require what's called a control group. Have you all heard this before? Yes. Of a control group? Do we put any sort of treatment on the control group? No. They're the control group that is your baseline that you're going to use to compare it to the ones who do get treatment. Now, because the way people are, if they think that nothing's happening, nothing's happening. There is a lot of times that treatments or individuals, experimental units will receive what's called a placebo. Have you all ever heard the word placebo? A placebo is like a fake treatment. But because, like psychologically, if we think we're a part of an experiment and we get some sort of treatment, we feel like we're a part of the experiment. So there may be some sort of change in you. If you know you're not getting the treatment, then there's some sort of psychology that goes along with it that might keep you from having any sort of change, or you might change a lot. I'm doing a terrible job of explaining this. But a placebo is a treatment that looks just like the real treatments in the study. A subject in the placebo group may find that the placebo treatment, literally like a sugar pill or something, resulted in complete or greatly improved state of being. This is interesting for our studies, and it is called the placebo effect. So if we think we're getting some treatment, we may start to change in some way. It's called the placebo effect. You didn't actually get any treatment, but huzzah, you're cured, something like that. In an experiment, it is important that each group be treated the same way, which is why we have a placebo, so that they receive some sort of pill, receive some sort of treatment. It is also important that individuals do not adjust their behavior because of the treatment they're receiving. Again, the placebo is helpful there. For this reason, many experiments use a technique called blinding. In a single blind experiment, have you ever heard these terms before? Single blind and double blind? Okay, basically what's happening in a single blind experiment, the subject or the experimental unit does not know which treatment he or she is receiving. So let's say, easiest thing to think about is like a diet pill, okay? Let's say you're in a study for a diet pill and they hand you a pill. You don't know if you got the actual medicine or if you got a sugar pill, which looks exactly like the medicine, but you don't know. A sugar pill is going to be like a piece of candy, okay? Like it's not going to do anything for you. It's got no medicine, no treatment. That's a single blind experiment because you, the subject, would not know what you're receiving. In a double blind experiment, neither the experimental unit nor the researcher in contact with the experimental unit knows which treatment the experimental unit is receiving. Which means that in my example of the diet pill study, if I didn't know what I was receiving, placebo or actual pill, the person dispensing it for me at the research center also does not know if me as the experimental unit is in the group that receives the sugar pill or the actual diet pill. That's called a double blind because you don't want them to act any different when giving the treatment, and you don't want the person receiving the treatment to act different knowing they're in one group or the other. That's called a double blind experiment. So single blind, the experimental unit does not know. Double blind means both do not know which of the treatment groups you're in. So let's see an example of a study and see if we can identify these different parts of the study. Example 1 says, Lipitor is a cholesterol-lowering drug made by Pfizer. In the collaborative diabetes study, which is the CARD study, the effect of Lipitor on cardiovascular disease was assessed in 2,838 subjects, ages 40 to 75, with type 2 diabetes without prior history of cardiovascular disease. In this placebo-controlled double-blind experiment, subjects were randomly allocated to either Lipitor for 10 milligrams daily, that's 1,428 people, or placebo, 1,410 people, and were followed for 4 years. The response variable was the occurrence of any major cardiovascular event, and Lipitor was shown to significantly reduce the rate of major cardiovascular events, 83 events in the Lipitor group versus 127 events in the placebo group. There were 61 deaths in the Lipitor group versus 82 deaths in the placebo group. This would be a study we could look at. The questions that we're going to discuss here is, what does it mean for this experiment to be placebo-controlled? What does it mean for the actual experimental design? How many groups do you have? 2. What does one of the groups get? The placebo. What does the other group get? So we are placebo-controlled because both groups are receiving some sort of, quote, treatment. Only one of them is an actual treatment. The other one is basically no change. But they're all receiving something from the researchers to maybe be a part of the study. What does it mean, second question, for the experiment to be double-blind?
The researcher and the experiment people don't know.
Correct. So both the subjects or experimental units and the researchers don't know which group it is. So when they roll up to the research center and say, I'm experimental unit number 5, somebody is going to look at number 5 and give them a bag. Now, that bag may have the placebo or it may have the whatever, but they always receive the same one. It's not just like rando. But we don't know, the researcher doesn't know until the very end when we draw conclusions which experimental unit got what. So it's double blind. The question, we'll highlight this one just to kind of get some highlighting on here. What is the population for which this study applies? Population. People ages 40 to 75, that's it? That's the only qualification?
Oh, with type 2 diabetes and no cardiovascular disease.
That is the population we're looking at. So we are basically only looking at Lipitor's ability to help lower the risk of cardiovascular event in middle-aged type 2 diabetes people with no prior cardiovascular disease. The population is very limited because we have to put some limitations on it. What is the sample? How many people did we use in the study? These subjects that we pulled, these 2,838 subjects are my sample, because this is only a small portion of the number of actual adults aged 40 to 75 with type 2 diabetes without a prior history of cardiovascular disease. So it's a subgroup, a sample. The next question asks, what are the treatments? Since we had 2 groups, we have 2 answers here. What are the treatments?
Lipitor reduced the rate of cardiovascular events.
So the treatments would be exactly what I'm giving the different groups. So one of them got Lipitor for 10 milligrams daily. What did the other group get? Placebo. Those are my 2 treatments. One of them is like not a treatment, right? Because it's a placebo, but that's the 2 treatments. The next thing we're going to look at is the response variable. This is like asking, what are we trying to study here? It does actually say it in this group, but what are we studying here? Yeah, the occurrence of any major cardiovascular event. That's what we're looking at to see if Lipitor had any sort of effect on. Is this response variable qualitative or quantitative? So qualitative is some quality or characteristic. Quantitative is numbers that we can add and subtract in some way and it's meaningful. Are we getting numbers out of this, or are we getting like a characteristic of people? We are actually getting characteristics of people. We are looking at the occurrence of any major cardiovascular event. So it's like a yes, no. Did you have a major cardiovascular event? Yes or no. That is a qualitative. If we all had, it would be quantitative, I guess, if it was like The rating of your cardiovascular events, like how severe was it? You could rate it on a scale of 1 to 10. That would make it quantitative, but since we're just seeing if this characteristic occurred, it's a qualitative variable. The actual steps for designing an experiment from start to finish would be number 1, identify the problem to be solved. We need to make sure we know exactly what we're asking for. The Lipitor study wasn't just like, does Lipitor lower the rate of cardiovascular event? It had to say in a specific population. Okay, that's a huge thing right there, is it has to explicitly state the response variable and the population. We want to know both. So we need to know the response variable and the population immediately so that we know this statement of the problem we're trying to study. Otherwise, it's too big. We have to narrow it down to a population and the response variable, one response variable. Number 2 is to determine the factors that may affect the response variable. So like if I'm just throwing this diet pill idea out there again, you would probably want age, you would probably want gender, you would probably want activity level. There's all these factors that would affect the response variable, which is probably, weight loss. So we have to think about all of those. As many of them as we can fix, we want to make sure we're talking to some professionals to actually fix these predetermined levels of these factors. We want to determine the number of experimental units, so that's your sample. In general, you're going to get as many as possible, as time and money allows. Step 4 is to determine the level for each factor. So we're going to control or randomize the different factors. As many as we can control, so that we only have one that varies, will be a better study. Then we're going to randomize our experimental units to the treatment groups. Step 5 is actually conducting the experiment, replicating the treatment for each experimental unit, collecting and processing your data. Step 6 is the big one. This is where we actually test the claims. So this is the subject of what we'll get to in inferential statistics, and after lunch we'll keep talking about it. There's the bell. Let's go through the design of an experiment that's called a completely randomized design. I love the name of it because it tells you exactly what's happening. It is completely randomized. It is when each experimental unit is randomly assigned to a treatment. It is a simplest type of experiment. So the 6 steps you'll see in this example are the 6 steps from above so that we can see what kind of questions we need to ask ourselves to make this completely randomized design. A farmer wishes to determine the optimal level of a new fertilizer on his soybean crop. We're going to design an experiment that will assist him. So the first thing we needed to do was identify the problem to be solved. The farmer wants to identify the optimal level of new fertilizer on his soybean crop. So the response variable will be what? What are we looking to study, to see a difference in?
Fertilizer.
That's what we're going to change. We're changing the fertilizer to see what? Optimal levels. In farming culture, that's called the yield. What is the population that we're working with? The soybean crop. Yeah, his soybean crop. That's our population. The second step is to determine the factors that affect the response variable. So some factors that affect the crop yield are fertilizer, which is what we're going to use as the treatments, precipitation, sunlight, method of tilling the soil, type of soil, plant type, and temperature. These are all things that we would need to take into consideration and control as much as possible so that we actually see the change due to the fertilizers, not due to accidentally something else, a lurking variable. In this experiment, we will plant 60 soybean plants, which means that those become our experimental units. Step 4 is to determine the level of each factor. Just in general, and your textbook listed out how to control all of these factors, but I didn't type them in because it was kind of repetitive. Can we control the amount of precipitation? No. Not really, because if it rains, it rains. But if we're looking at one farmer's field, they're all going to get rained on about the same, yeah? And we can control the amount of watering we do. So precipitation will be controlled. How about sunlight? Can we actually control that? No, but again, they're all in the same field, so they're going to get approximately the same amount of sunlight. The method of tilling the soil, we make sure that we're preparing the soil in the same way so that that doesn't happen to be some sort of lurking variable. The type of soil we're going to use, we really can't control, but again, when it's one plot of land, we can generally say the soil is going to be the same. The plant type, we already know the differences in, and we'll keep them to be the same type of plant, a soybean plant. And temperature, we can't really control the temperature, but again, since they're in the same plot, we can say they all have the same temperature. So every factor has been controlled except for fertilizer, because that's what we're giving treatments to. We're going to split these 60 soybean plants into 3 groups. Treatment A is no fertilizer, which means treatment A is our what group? Control group. There's no placebo here because you don't have to fake a plant into thinking it's getting fertilizer. You just don't give it fertilizer. Treatment B will be, I accidentally deleted the number. It will be 2 teaspoons. So sorry, 2 teaspoons of fertilizer per gallon of water every 2 weeks. And treatment C will be 4 teaspoons of fertilizer per gallon of water every 2 weeks. It is very specific in how much and in what way we are doing these treatments. When we get to step 5 with the replication, that means we are going to randomly assign each plant to a treatment group. To do this, first we're going to number the plants from 1 to 60 and randomly generate 20 numbers in some way that we did last time. Maybe with our calculator, maybe with a table of random numbers. These plants corresponding to these numbers will get treatment A. Then we're going to take the remaining plants, number them 1 to 40. Again, randomly generate 20 numbers in one of the ways we learned last time. And then the ones left over after that will get treatment C. We'll collect the data at the end of the growing season and determine the crop yield for each plant. Step 6 is looking at our data and doing inferential statistics. So we'll look at any differences in yield among the 3 treatment groups. So visually, and what I'm going to ask you to draw for me is some sort of flow chart that represents this experiment. We start with our population. We have our 60 soybean plants, or sorry, this is our sample, 60 soybean plants. We're going to split them into 3 groups. So we have treatment group A, which is 20 random plants. We'll have treatment group B, which is again another 20 random plants, making sure we have that word random there to help us remember that we have to do a random sample in one of the methods we learned last class. And then treatment C, we have our 20 remaining plants. That was horrible handwriting. Let me try that again. Now, each of these groups, A, B, and C, will then receive a treatment. So we have treatment A. What was treatment A again? The control, which was no fertilizer. Treatment B was how many teaspoons? 2 teaspoons per gallon of water every 2 weeks. Treatment group C was 4 teaspoons. So we have the population split into the 3 sample groups, or the 3 treatment groups. The treatments. After we do the treatments, we're going to have our results by comparing the yields. Notice we're comparing the yields against all 3 together. That's important for later. All 3 of them get compared together so that we can see if there's any benefit or what the benefit is to no fertilizer, 2 teaspoons of fertilizer, or 4 teaspoons of fertilizer. Does this study design make sense? Randomly split them into groups, assign a treatment to each, compare all together. Check. That's called a completely randomized design. The second type of design we can have is called a matched pairs design. Again, I love the way we've named these things because they tell you exactly what to do. In a matched pairs design, it's an experimental design in which the experimental units are paired up. The pairs are selected so that they are related in some way. That is the same person before and after an experiment, twins, a husband and a wife, same geographical location, and so on. They need to be matched by some characteristic or some relationship. There are only 2 levels of treatment in a matched pairs design because we're comparing basically a before and after. We are comparing one twin to the other, and on and on and on. So let's see what a matched pairs design flow of an experiment might look like. An educational psychologist wants to determine whether listening to music has an effect on a student's ability to learn. Design an experiment to help the psychologist answer the questions. So we get a group of kids. How could we match these students to maybe make this a really good study? If we're trying to see their ability to learn, what is maybe one thing we should match them on? Grades. We're going to call that IQ. The other thing we're going to match them on is gender, just to see if, again, it helps us control to see if there's a difference in males and females. Match them. Then, again, key word here is we are going to randomly assign a student from each pair to a treatment. We're going to administer the treatment to one of the two pairs, which is 2 hours of study time with music or 2 hours of study time without music, and give them an exam, a short quiz over the topics they were studying, to each matched pair. Then for each matched pair, we're going to compute the differences in score on the exam. It's like comparing the yields. So this one is different where we're not taking large groups of plants and separating them into treatment groups. We're going to make sure each person, each experimental unit has a pair that they're related to in some way so we can see the differences in them. Now this is used a lot. Twin studies are very important actually because it keeps genetics the same, which is kind of interesting. But these are usually done in a before and after. So we're going to highlight those words up there, before and after a treatment. You'll see them called pre and post tests, which are just before and after the treatment on the same individual. So that's a matched pairs design. You're matched basically with yourself if you're doing that sort of study. Matched pairs design. Love it because it's in the name. You match your pairs. It's a matched pairs design. The last design of a study that we're going to talk about today is a randomized block design. Randomized is going to feel a lot like the simple randomized design. This is a randomized block design. This occurs when we are grouping together similar or homogeneous experimental units and then randomly assigning the experimental units within each group to a treatment called blocking. Each group of homogeneous individuals is called a block. A randomized block design then takes these blocks, these two groups, and basically does a simple randomized design on each group. Within each block, the experimental units are randomly assigned to the treatments, the same way we did before. So let's go back to our plants. Let's suppose that these 60 soybean plants are actually two varieties of soybean plants. We have 30 of them that are ChemGrow soybean plants. I guess that's a type of soybean plant. And 30 Pioneer soybean plants. Again, a type, a brand, not sure. Didn't Google that, I'll be honest. And the two varieties have naturally different yields. Let's say ChemGrow already has a higher yield than Pioneer. We wouldn't want to compare them to each other and put it just in a simple group because naturally the ChemGrows are going to have more. So what we do is we separate them into two blocks, the two types of plants, and within those two types, then we do the completely randomized design. So here's what our design might look like. We have our 60 soybean plants into two blocks. We have our 30 ChemGrow, and we're going to have our 30 Pioneer. We're separating them first by type because there are naturally differences in these types that we need to weed out as part of the problem. But from each of those 30, we're going to assign 10 randomly to different treatments. So we're going to choose 10 random plants to get the no fertilizer. We're going to have 10 random plants get the 2 teaspoons of fertilizer. Then we're going to have the last 10. It says remaining. They're still technically random. And that's 4 teaspoons. And that's all with the ChemGrow. And we're going to look at the ChemGrow and we're going to compare yields. Then we're going to go over to our plot of land that has all the Pioneer soybeans in it and assign 10 random of those plants to have no fertilizer. 10 random of those plants to have 2 teaspoons a fertilizer per gallon per week, however we decided. And then the 10 remaining will get the 4 teaspoons, a lot of fertilizer. Then we're going to compare the yields of all the Pioneer plants. Notice, did I draw any connecting dots between the 2 boxes that say compare yields? No. We already know that ChemGrow might already produce more. So we don't want to accidentally draw a conclusion about our fertilizer that was due to just the fact that like ChemGrows already grow higher, more. We have to make sure that we're blocking these by type when there's a significant difference in those types so we can actually see if the fertilizer affects. And maybe it does really, really well with the Pioneer and not really well with the ChemGrow. Save your money, don't buy the fertilizer for half your plants. These are the sort of things we want to see when we block our design before we randomize. Splitting into multiple groups when their groups are different enough. And it says all of this right here, what I just said out loud. Okay, so your homework today is just 3 questions if you look at it, and it's going to go through an experiment, and it's going to have you identify like population, sample, response variable, treatments, all of that stuff, and then it wants you to draw a designed experiment for it. So you're going to draw some sort of like flow chart, bubble chart for these. I'm not providing an answer key for this one. So what I want you to do is work by yourselves. And when you think you have a good answer, talk to your study group and just discuss if your treatment style, if your design makes sense. There's more than one correct way to draw these bubbles. Just make sure it follows the rules, if that makes sense. Okay, you have the rest of the class. Oh, we have lots of time to keep working on. I'm still recording.